7 Chapter 3 Plant Kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker 1969, where in this suggestion the five kingdom classification Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and the members of the Monera and Protista having cell wall have now been excluded from Plantae through earlier classification, placed them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe Plantae under algae, bryophyte, pteridophyte, gymnosperm, and angiosperm. Let us also look at classification within angiosperm to understand some of the concerns that influence the classification system. The earliest system of classification used only gross superficial morphological characters such as habit, color, number, and shape of the leaves, etc. They were based mainly on vegetative character or on the endoxium structure. System given by lineage. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to the vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative character are more easily affected by the environment as against the natural classification system developed which were based on natural affinities among the organisms and considered not only the external features but also internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on the evolutionary relationship between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help to resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers based in all observable characteristic number and code assigned to all the characters and the data and then proceeds. In this way, each character is given equal importance and at the same time hundred of characters are considered. Cytotaxonomy that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy that uses the chemical constituents of the plant that resolve confusion are also used by taxonomists these days. 3.1 Algae. Algae are chlorophyll bearing simple thalloid, autotrophic, and largely aquatic, both fresh water and marine organisms. They occur in a variety of other habitats, moist stones, soils, and wood. Some of them also occur in association with fungi, lichens, and animals, example slothware. The form and size of algae is highly variable. Figure 3.1. The size ranges from the microscopic unicellular forms like Chlamydomonas to colonial forms like volvox and the filamentous forms like Elothrix and Spirogyra. A few of the marine forms such as scalps form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual method. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores. The most common being the zoospore. They are flagellated, motile, and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Chlamydomonas, or non-flagellated, non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes is similar in size as in species of Chlamydomonas is termed as angiosperm. An isogamous fusion between one large non-motile static female gamete and a smaller motile male gamete is termed as oogamous. Example Volvox fucus. Figure 3.1 Algae. A. Green algae Volvox Chlamydomonas cara. B. Brown algae Laminaria fucus decuota. Third, red algae Porphyra polysphonia. Algae are useful to man in the variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compounds which form the basis of the food cycle of all aquatic animals. Many species of porphyra, the basis of the food cycles of all aquatic animals. Many species of porphyra, laminaria, and sargassum are among the 70 species of marine algae used as food. Certain marine brown and green red algae produce large amount of hydrocolloids, water-holding substance. Example: Algin brown algae and Cara green red algae, which are used commercially. Agar, one of the commercial products obtained from Galidium and Crassularia, are used to grow microbes and in preparation of ice cream and jellies. Chlorella, a unicellular algae rich in protein, is used as food supplement. Even by space travel, the algae are divided into three main classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. 3.1.1 Chlorophyceae. The members of the Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, and filamentous. They are usually grass green due to dominance of pigment chlorophyll A and B. The pigment are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped. In different species, most of the members have one or more storage bodies called prenoids located in the chloroplast. Prenoid contain protein besides starch. Some algae may store food in the form of oil wrapped. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative production usually take place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangia. Sexual reproduction show considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous, and or ogamous. Some commonly found green algae are Chlamydomonas, Volvox, Ulothrix, Spirogyra, and Cara. Figure 3.1a. 3.1.2 Phaeophyceae. The members of Phaeophyceae are brown algae are found primarily in marine habitat. They show great variation in size and form. They range from simple branched filamentous form ectocarpus to profusely branched forms are represented by kelps which may reach a height of 100 meters. They possess chlorophyll, AC, carotenoid, and xanthophylls. They vary in color from olive green to various shades brown depending upon the amount of xanthophyll pigment fuchsanthin present in them. Food is stored as complex carbohydrate which may be in form of laminin and mannitol. The vegetative cell have a cellulosic wall usually covered on the outside by gelatinous coating of algin. The protoplast contain, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a hold fast and has a stalk. This type and leaf-like photosynthesis organ. The frond vegetative reproduction take place by fragmentation. Asexual reproduction in most brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the oogamous species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common forms are Ectocarpus, Dichota, Laminaria, Sargassum, and Fucus. Figure 3.1b. 3.1.3 Rhodophyceae. The members of Rhodophyceae are commonly known as red algae because of the predominance of the red pigment. Are phytoerythrin in their body. Majority of the red algae are marine with greater concentration found in the warmer areas. They occur in both well-hidden regions close to the surface of water and also at great depth in ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile. Gamete sexual reproduction is oogamous and accompanied by complex post-fertilization development. The common members are Polysiphonia, Porphyra, figure 3.1c, Gracilaria, and Galidium. 3.2 Bryophyte. Bryophyte include the various mosses and lavort that are found commonly growing in moist shaded areas in the hills. Figure 3.2. Figure 3.2 Bryophytes. A. Liveworts, Marchantia. B. Female thallus. B. Male thallus. Mosses. C. Funaria, gametophyte, and sporophyte. D. Spagum, gametophyte. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are depend on water for sexual reproduction. They usually occur in damp, humid, and shaded facilities. They play an important role in plant succession on drained rock soils. The plant body of bryophyte is more differentiated than that of algae. It is a thalamus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem, or leaves. They may possess root-like, leaf-like, or stem-like structure. The main plant body of the bryophyte is haploid. It produces gametes, hence called gametophyte. The sex organ is in bryophyte are multicellular. The male sex organ is antheridium. They produce biflagellate antherozoids. The female sex organ called archegonium flask-shaped and produce a single egg. The antherozoids are released into water where they come in contact with archegonium and antherozoid fuse with the egg to produce the zygote. Zygote do not undergo reproduction division immediately. They produce a multicellular body called a sporophyte. Sporophyte is not free living but attached to the photosynthetic gamete and derives nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce haploid spore. These spores germinate to produce gametophyte. Bryophyte is generally are of little economic importance, but some mosses provide food for herbivorous mammals, birds, and other animal species of Shagnum. A moss provide peat that have long been used as fuel and as packing material for transshipment of living material because of their capacity to hold water. Mosses, along with lichen, are the first organism to colonize rock and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the important impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. 3.2.1 Lavort. Lavort grow usually in moist, shady habitats such as bank of streams, marshy ground, damp soil, bark of fruit trees, and deep in the woods. The plant body of liverwort is thalloid. Example: Marchantia. The thallus is dorsiventral and closely pressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stream-like structure. Asexual reproduction liverworts take place by fragmentation of thalli or by the formation of specialized structure called gamia. Syngamia. Gamia are green, multicellular asexual buds which develop in small receptacles called gamma cups located on the thalli. The gamma become detached from the parent body and germinate to form new individual during sexual reproduction. Male and female sex organ are produced either on the same or on the different thalli. The sporophyte is differentiated into food, seeds, and capsules. After meiosis, spores are produced within the capsule. These spores germinate to form free living gametophyte. 3.2.2 Mosses. The predominant stage of the life cycle of a moth is the gametophyte, which consists of two stages. The first stage is protonema stage, which develops directly from a spore. It is creeping green branch and is frequently lemontinous stage. The second stage is leafy stage, which develops from the secondary protonema and as lateral bud. They consist of upright, slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizomes. This stage bears the sex organ. Vegetative reproduction in mosses by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organ antheridia and archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into the sporophyte. consisting of a food seta and capsule. The sporophyte in mosses is more elaborate than that in liverwort. The capsule contain spores. Spores are formed after meiosis. The meiosis have an elaborate mechanism of spores dispersal. Common example of meiosis are funaria and polytrichum and sphagnum. Figure 3.2. 3.3 Pteridophyte. The pteridophyte include horsetail and ferns. Pteridophytes are used for medicine purpose and as soil blinders. They are also frequently grown as ornamentals. Evolutionary, they are the first terrestrial plant to possess vascular tissue, xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophyte the dominant place in their life cycle is gametophytic plant body. However, in pteridophyte life main plant body is sporophyte, which is differentiate into true body, true root, stem and leaves. Figure 3.3. These organs possesses well differentiated vascular tissue. These leaves in pteridophyte are small microphyll as in Selaginella or large macrophyll as in fern. The sporophyte bear sporangia that are subtended by leaf-like appendages called spirophytes. In some cases, spirophylls may form distinct compact structure called strobili or cones. Selaginella equistem, the sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous small but multicellular. Figure 3.3 Pteridophyte. Selaginella. B. Equistem. C. Fern. D. Salvinia. Free living, mostly photosynthetic thalloid gametophyte called prothallus. These gametophyte require cool, damp, shady places to grow because of this specific restricted requirement and the need for water for fertilization. The spread of living pteridophyte is limited and restricted to narrow geographical region. The gametophyte bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of anthera. The male gamete release from the antheridia to the mouth of archegonium. Fusion of male gametes with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produce a multicellular well differentiated sporophyte, which is dominant phase of the pteridophyte. In majority of the pteridophyte, all the spores of similar kind. Such plant are called homosporous genera like Selaginella and Salvinia, which produce two kinds of spores. Macro means large and micro means small. Spores are known as heterosporous. The megaspore and microspores germinate to give rise to female and male gametophyte respectively. The female gametophyte in these plants are retained on the parent sporophyte for variable period. The development of the zygote into young embryo take place within the female gametophyte. The event is a precursor to the seed habit, considered an important step in evolution. The pteridophyte are further classified into four classes: Celosodia, Siltum, Lycospida, Selaginella, Lycopodium, Phanosidia, Equistem, and Pteridosidia, Trioteris, Teres adenatum. 3.4 Gymnosperm. The gymnosperm naked sperma seeds are plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post fertilization are not covered, are naked. Gymnosperm include medium sized trees or tall trees and shrubs. Figure 3.4. One of the gymnosperm that giant redwood tree sequoia is one of the tallest tree species. The roots are generally taproot. Roots in some genera have fungal association in the form of mycorrhiza pinus, while in some others caicus. Small specialized roots called caroloid roots are associated with nitrogen fixing cyanobacteria. The stems are unbranched cycas or branched Picus cadrus. The leaves may be simple or compound. In Caicus, the pinnate leaves persist for a few years. The leaves in Gymnosperm are well adapted to withstands extreme of temperature, humidity, and wind. In conifer, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperm are heterosporous. They produce haploid microspore, megaspore. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form legs or compact strobili or cones. The strobili bearing microsporophyll and microsporangia are called microsporangiate or male strobili. The microspore develop into a male gametophyte generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophytes called a pollen grain. The development of pollen grain take place within the microsporangia. The cones bearing megasporophyll by ovules or megasporangia are called megasporangiate or female strobili. The male or female cones or strobili may be born on the same tree, Pinus. However, in Cycas, male cones and megasporophyls are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucleus. The nucleus is protected by envelope of the composite structure is called an ovule. The ovule are born on megasporophyll, which may be clustered to form a female cone. The megaspore mother cell divide meiotically to form four megaspores. One of the megaspore enclosed within the megasporangium develop into a multicellular female gametophyte that bears two or more archegonium or female sex organ. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophyte and pteridophyte, is gymnosperm, and the male and the female gametophyte do not have a independent free living existence. They remain within these sporangia, retain on the sporophyte. The pollen grains released from the microsporangium. They are carried in air current and come in contact with the opening of the ovule born on megasporophyll. The pollen tube carrying the male gamete grow out toward archegonia in the ovules and discharge their content near the mouth of the archegonia. Following fertilization, zygote develop into an embryo and ovule into seed. These seeds are not covered. 3.5 Angiosperm. Unlike the gymnosperm, where the ovules are naked, in the angiosperm or flowering plant, the pollen grain and ovules are developed in a specialized structure called flower. In angiosperm, the seeds are enclosed by fruit. The angiosperm are an exceptionally large group of plants occurring in wide range of habitat. These range in size from tiny, almost microscopic Wolfia to tall tree eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicine, and several other commercially important products. They are divided into two classes: the dicotyledon and the monocotyledon. Figure 3.5. The dicotyledons are characterized by having two cotyledons and their seeds, while the monocotyledon have only one. The male sex organ in a female is the stamen. Each stamen consists of a slender filament with an anther at the tip. The anther, following meiosis, produce pollen grain. The female sex organ in a flower is a pistil or the carpel. Pistil consists of an ovary enclosing one of the many ovules. Within the ovule are present highly reduced female gametophyte termed embryo sacs. The embryo sac formation is preceded by meiosis. Hence, each of the cell of an embryo sac is haploid. Each embryo sac has a three-celled egg apparatus, one egg cell and two synergists, three antipodal cells and two polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain after dispersal from the anther are carried by wind or various other agencies to the stigma or of a pistil. This is termed as pollination. The pollen grain germinate on the stigma and the resultant pollen tube grow through the tissues of the stigma and the style reach the ovule. The pollen tube enters the embryo sac where two male gametes are discharged. One of the male gametes fuses with the egg to form zygote syngamy. The other male gamete fuses with the diploid secondary nucleus to produce a triploid primary endosperm nucleus pen. Because of the involvement of tube fusion, this event is termed as double fertilization, an event unique to angiosperm. The zygote develops into an embryo with one or two cotyledon and the pen develop into endosperm which provide nourishment to the developing embryo. This Ncert and the antibodal degenerate after fertilization. During these events, the ovules develop into seeds and ovaries develop into fruit. The life cycle of the angiosperm shown in figure 3.6. Figure 3.6 life cycle of an angiosperm. 3.6 Plant life cycle and alternation of generation. In plant, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produce gamete by mitosis. This plant body represent a gametophyte. Following fertilization, the zygote also divide by mitosis to produce a haploid sporophyte plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of the generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant group as well as an individual representing them differ in the following pattern. First, sporophytic generation is represented only by one-celled zygote. There is no free-living sporocytes. Sporophyte meiosis in the zygote result in the formation of haploid spores. The haploid spore divide mitotically to form the gametophyte. The dominant photosynthetic phase in such plant is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as Volvox, Porogyra and some species of Chlamydomonas represent this pattern. Figure 3.7a. Second, on the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by single to few celled haploid gametophyte. This kind of life cycle is termed as diplontic, and alga, fucus, sep represent this pattern. Figure 3.7b. In addition, all seeds bearing plants, gymnosperms and angiosperm follow this pattern with some variation, wherein the gametophytic phase is few to multi-celled. Third, bryophyte and pteridophyte interestingly exhibit an intermediate condition, haplodiplontic. Both phases are multicellular, however they differ in their dominant phase. Plant kingdom include algae, bryophyte, pteridophyte, gymnosperm, and angiosperm. Algae are chlorophyll-bearing, simple thalloid, autotrophic, and largely aquatic organisms. Depending on the type of pigment process and the type of stored food, algae are classified into three classes, namely chlorophyll, phyophyceae, and rhodophyceae. Algae are usually reproduced vegetatively by fragmentation, asexually reproduced by formation of different types of spores, and sexually by formation of gametes which may show isogamy and oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like, prostate and erect and attached to the substratum of rhizobium. They possess root-like, cleft-like and step-like structure. The bryophytes are divided into liverworts and mousses. The plant body of liverwort is thyroid and dorsiventral, whereas mousses have upright, slender axis bearing spirally arranged leaves. The main plant body of bryophyte is gamete producing and it's called gametophyte. It bears the male sex organ called antheridia and the female sex organ called archegonia. The male and female gamete produce fuse to form a zygote which produces a multicellular body called sporophyte. It produces haploid spores. The spores germinate to form gametophyte and pteridophyte. The main plant is the sporophyte, which is differentiated into the two roots, stem and leaf. These organs possesses well differentiated vascular tissues. The sporophyte bears sporangia which produce spores. The spores germinate to form gametophyte which require cool damp places to grow. The gametophyte bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of male gametes to archegonium where zygote is formed after fertilization. The zygote after a sporophyte produce a sporophyte. The gymnosperm are the plant which ovules are not enclosed by any ovary. After fertilization, the seed remain exposed and thereafter these plants are called naked seeded plant. The gymnosperm produce microspores and megaspore, which are produced in microsporangia and megasporangia bone of saprophyll. The saprophylls, microsporophylls, and megasporophylls are arranged spirally on axils to form male and female cones respectively. The pollen grain germinate the pollen tube, release the male gamete into the ovule, where in the fuse with the egg in archegonium. Following fertilization, the zygote develops into embryo and the ovules into seed. In angiosperm, the male sex organ, stamen, and the female sex organ, pistil, are born in a flower. Each stamen consists of filament and anther. Anther produce pollen grain, male gametophyte. After meiosis, the pistil consists of an ovary enclosing one to many ovules. Within the ovule is the female gametophyte or embryo sac, which contain an egg cell. The pollen tube enters the embryo sac, where two male gametes are discharged. One male gamete fuses with egg cells and gamete, and the other fuses with diploid secondary nucleus. Triple fusion. This phenomena has two fusion is called double fertilization, and the unique to angiosperm. The angiosperm are divided into two classes: the dicotyledon and the monocotyledon. During the life cycle of any sexually reproducing plant, there is an alternation of the generation between gamete producing haploid gametophyte, the spore producing diploid sporophyte. However, different plant group as well as an individual may show different patterns of life cycle, haplontic, diplontic, or intermediate.